Hello to all my dear lovely students. I hope you all are doing fine. So once again I welcome you on this amazing platform of Physics Wallah where you are studying in excellence batch. So far we have completed the first lecture of the second chapter class 12, that is sexual reproduction in flowering plants. Okay, today we are going to start with the lecture number 2. In our lecture number 1, we have discussed about the male reproductive androecium and where in androecium actually microsporogenesis takes place for the production of haploid male gametophyte microspore, also known as pollen grain. I hope you all have gone through that lecture and you all have understood the actual concept behind the formation of pollen grain, that is the male gametophyte. Today, we are going to understand the details of pollen grain, that is the male gametophyte, along with the structure of female reproductive organ, that is gynoecium. I hope you all are ready. So, chalo. Why to wait for? Let's directly start our today's session with the male gametophyte, that is pollen grains. Usually beta, pollen grains are spherical structures which having an average size of 25 to 50 microns. Right? So this is very important. The first line itself, that pollen grains are haploid microspores that develop after the meiosis in the diploid microspore mother cell within the anther of the stamens. These pollen grains are spherical in structure with an average size of 25 to 50 microns. Right? Structurally, if I talk about beta, pollen grains are two layered bound structure. Both the layers together they are known as sporoderm. The two layers are inner is intine whereas outer is exine. The inner intine layer is continuous beta like this. So the inner intine layer is continuous, made up of cellulose and pectin. So inner is intine, which is continuous pectocellulosic. This intine layer, my dear students, surrounds the cytoplasm along with a nucleus. Right? Outer to intine is present the discontinuous hard layer called exine. Discontinuous means what? That there are some openings which are present in the exine layer. So this is exine. The inner is intine. Together they are known as sporoderm, and this is discontinuous, hence you can see presence of germ pores. What are there? Germ pores are there. Correct? Now, this exine is made up of sporopollenin. Sporopollenin is the chemical which is secreted by tapetum layer. Sporopollenin, my dear students, it is said to be the hardest chemical found ever because it cannot be degraded with the help of any alkali, any other chemical, temperature, etc. So sporopollenin is the hardest substance known which cannot be degraded. As a result, due to the deposition of sporopollenin, pollen grains are also non-degradable and they persist for years after year. Normally, the study of pollen grain is known as palynology. However, what am I saying? However, due to the deposition of sporopollenin on the exine, it becomes what? It becomes indestructible. Because it is indestructible, it persists for years and years. Therefore, some scientists, archaeologists, they use pollen grains to study archaeology, to study the history, to study the extinct plant species. Plants have extincted, but their pollen grains might still persist in today's environment. So scientists, the archaeologists, they use pollen grains to study the fossil records. Hence, when you use pollen grains to study fossil record, that study is known as geopallinology. Right? So let's write this point also. What we are going to write? Due to the deposition of sporopollenin, which is an indestructible chemical, pollen grains are also used for studying fossils, right? For studying fossils, therefore it is also, and using pollen grains for studying fossil, this is known as geopallinology. I hope both the terms and the difference is clear. Pallinology and geopallinology. Very good. Coming to the next point, this is how beta the haploid microspore looks like. This is how pollen grain actually looks like. Now, this pollen grain, if wants to germinate on the stigma, then what happens? Through the germ pore, intine bulges out to form the pollen tube. Correct? So what I am going to tell you, now see, this is your intine which bulges out through the germ pore to form the pollen tube. So basically, what is pollen tube? It is actually the bulged intine. Where the bulging is taking place? Through the germ pore. Now beta, intine cannot break through exine. Exine is indestructible due to the deposition of sporopollenin. So from where intine should come out? From where pollen tube should be formed? So formation takes place through the germ pore. Now as soon as the pollen tube comes out, the cytoplasm comes at the tip. So tip is then occupied by active cytoplasm. So this is your microspore. You know, one more point I wrote beta, that this pollen grain is actually having a nucleus. This nucleus undergoes mitotic division. This is a haploid nucleus which undergoes mitotic division to produce two irregular sized cells, the larger called vegetative cell and the smaller spindle shaped called generative cell, which floats in the cytoplasm of vegetative cell itself. Correct? So just a second. Okay, so that point I have written later on, but here only I am going to explain you the nucleus which is there inside the haploid cell undergoes nucleus of haploid pollen grain beta undergoes mitotic, asymmetric mitotic division to produce two cells, right? There is a larger called vegetative cell, also called as tube cell, and the smaller called generative cell, right? This generative cell is small and spindle shaped. Small and spindle shaped, which floats in the cytoplasm of spindle shaped, which floats in the cytoplasm of larger vegetative cell. Correct? Yes or no? Like this, if this nucleus undergoes division, chalo see, now let's see how it will look like. So this is the larger vegetative cell, in the cytoplasm of which there is smaller generative cell. So this is vegetative cell and this is spindle shaped smaller generative cell. This is how your male gametophyte looks like. This is what pollen grain is. Now one more point I am going to tell you related to exine. Beta, exine is discontinuous, you know. It is deposited with sporopollenin. Now exine of different pollen grain species are of different shapes. Right? As a result, exine is also used to study the pollen grains of different species. Exine, they are sculptured. There are some designs on exine due to the deposition of different chemicals in different forms. As a result, the structure of exine is also used to study the pollen grains of different species. So this point also you can remember. Now, coming ahead, now you have seen the two types of cells which are produced due to asymmetric mitosis, correct? Now next topic in pollen grain is pollen allergy. Beta, usually those plants whose pollen grains are dispersed through wind, anemophilous plants which we say, their pollen grains sometimes can cause allergy if you inhale them. Some people, they are allergic to some pollen grains. If they intake, they might start sneezing, coughing, itching, redness. In extreme case, it can also cause troubles like bronchitis or even asthma. The three common species of whose pollen grains are known to cause severe allergies are Parthenium, Chenopodium and Amaranthus. Correct? Shall I write the points? What is pollen allergy? Pollen allergy means affliction in the respiratory tract due to inhalation of pollen grain. Or you can simply say some wind pollinating plants, anemophilous plants, some wind pollinating anemophilous plants, their pollen grains, comma, their pollen grains can inflammate the respiratory tract. Can inflammate the respiratory tract, causing breathing issues like bronchitis, asthma, causing bronchitis, asthma. In small cases, it leads to coughing, sneezing and irritation in eyes, irritation in the skin. These are some of the symptoms of pollen allergy. The most common species whose pollen grains are known to cause allergy are Parthenium. This is also known as carrot grass or Congress grass. Parthenium. Beta, Parthenium is an exotic species of a country. In our country, Parthenium was not there. It was unknowingly, by chance, introduced in our country and it became a terrible terror. So it is an actually exotic species which is now found in every single part of our country. Even outside your house, you can find Parthenium. It is now called a wild weed, which is growing everywhere. You cut it, again it will come out. Without any extra supervision, Parthenium grows by itself. And the problem with Parthenium is, beta, it consumes lot amount of water. And it releases certain chemicals. The roots of Parthenium, they release chemical and they absorb lot amount of water, because of which the area near Parthenium is not fit for growing other crops. because other crop will face problem with the chemicals released by them, plus there will be no water left. So Parthenium thus is an exotic weed that creates lot of problem in our country. Correct? Apart from Parthenium, the second is Amaranthus. The pollen grains of Amaranthus are also irritating. Third example which you have to remember is Chenopodium. So please remember these three examples given in your NCERT and you should know them. Next is pollen viability. Pollen viability means the time span for which pollen grains remain alive. Like suppose they are discharged from the anther. Now 3 days they may take to land on stigma to germinate. Some pollen grains may take 5 days, 1 week, months to land on stigma. But they may not be alive for so many days. So what is pollen viability? Actually, the lifespan of pollen grains once it is discharged from the anther till the time it lands on the stigma, successfully lands on the stigma, that is known as pollen viability. So what is pollen viability, students? It is basically the lifespan of pollen grain, which ranges from the time from the release of pollen grain from the anther till the time it lands on the stigma. Pollen viability, lifespan of pollen grain, that is the time period for which pollen grains remain alive. For which pollen grain remains alive is known as pollen viability, and it is variable from species to species. Like for example, for rice, wheat, their pollen grains are only viable for 30 minutes. That means if pollen grains are shed from the anther and if within 30 minutes they do not successfully land on stigma, then those pollen grains are dead, of no use, you cannot do anything. So their viability, their lifespan is only of 30 minutes. Similarly, like the pollen grains of the family members Leguminosae, Solanaceae, Rosaceae, their pollen grains are viable for several months. Then members of, here I am writing the other example. This is example number 1. This is example number 2. Members of family Leguminosae, Solanaceae and Rosaceae, their pollen grains are viable for several months. Correct? Yes or no? Will you remember these two statements? Pollen allergy and pollen viability, very, very important. Correct? Now beta, sometimes pollen grains are also consumed as a source of energy. In markets, pollen tablets are available which are taken in by the athletes. Not by us, because we do not need so much of energy. These pollen grains are very rich in energy. Beta, they are carrying the male gamete. Right? The structure which is carrying the male gamete has to be highly energetically expensive. Right? So, these pollen grains, they are even used in the form of syrups, in the form of tablets by the athletes to increase their performance. Right? So, pollen grains are also consumed in the form of tablets, in the form of syrups by the athletes to improve their performance. So this thing also you can write pollen consumption. What will you write for pollen consumption? Pollen grains of some species are used as tablets taken by the athletes. Pollen grains as tablets and syrups are taken up by the athletes. Are taken up by the athletes to improve their performance. Right? So pollen consumption, pollen grains as tablets and syrups are taken up by the athletes to improve their performance, as pollen grains of some species are highly energetically strong. Now, next topic is pollen banks. Pollen banks represents those areas where we can preserve, where we can conserve the pollen grains in liquid nitrogen at minus 196 degrees Celsius. In that case, you can conserve pollen grains for years. They will remain alive even after, suppose, rice pollen grain. Normally, they are only viable for 30 minutes. But suppose I need the same pollen grain for my next year crop. But naturally pollen grains will die in 30 minutes. So how can I use it for my next year? What will I do? I will conserve those pollen grains. I will let those pollen grains undergo sleep mode by keeping them under liquid nitrogen at minus 196 degrees Celsius. I will preserve it for 1 year. And once I will need it, I will take out them. I will give them heat shock. The again inactive cell will become active and I can use it for my crops. So that is called as pollen banks. As and when required, pollen grains can be, not preserved word is wrong, I'm sorry. It should be conserved. So pollen grains are conserved at liquid nitrogen in liquid nitrogen at minus 196 degree Celsius. Correct? Like you save your money. Suppose today I am earning more than what I require. So will I throw the extra money? No, of course I will store it in my bank like SBI, RBI, etc. Similarly, when you may have extra pollen grains, farmers, or suppose if they want to conserve them for their next season use, and suppose viability is of lesser time. So again, you can store those in the pollen banks. Correct, you don't throw them, you store them. So what are pollen banks? These are the places to conserve pollens for future use. So pollens over here, they can be conserved for future use. What will you do? You will keep them in liquid nitrogen at minus 196 degrees Celsius temperature. At this low temperature, we can conserve pollen grains and use it as and when required. I hope all the terminologies, pollen viability, pollen banks, pollen energy, and pollen tablets are clear to every one of you. If yes, next topic, which I have already told you, the cells, the nuclei of pollen grain divides by mitosis to produce two irregular shaped cells, vegetative and generative. Vegetative is smaller, vegetative is larger and generative is smaller, which floats in the cytoplasm of vegetative cell. So you have already seen it with the help of the diagram also. Therefore, I am proceeding further. Generative cell is the smaller cell. That floats in the cytoplasm of vegetative cell. Correct? Next point, see beta, this point is very, very important. Please see this point. This point says, the next point says that 60% of angiospermic family, majority of the families, they shed their pollen grains at this two-celled stage. Correct? At two-cell stage, that means In 60% of the angiospermic family, the dehiscence of anther takes place when pollen grains are having two cells, generative and vegetative. However, in remaining 40% of the angiospermic family, what happens? This generative cell further undergoes asymmetric division. Again, asymmetric division takes place. Mitosis takes place to produce two non-motile amoeboid shaped, irregular shaped male gametes. So this is your generative cell which undergoes asymmetric mitosis to produce two amoeboid, irregular shaped, non-motile male gametes. So now how many total cells will be there in pollen grain? 3 cells, 1 vegetative and 2 male gametes. So in 40% of the angiospermic families, the generative cell within the anther, still there in the anther, within the anther they divide to produce 2 male gametes, and thus in 40% of the families, dehiscence takes place at 3-celled state. A pollen grain with 3 cells is a complete mature male gametophyte. So pollen grain at 3-celled condition, at 3-celled condition, my dear student, is referred to as mature completely, because male gamete is produced. Pollen grain is the male gametophyte, male gamete carrying body. So once male gamete is produced, it is a mature. Now you can call pollen grain as male gametophyte, as a mature male gametophyte. Now you can call this. In 60% of the angiospermic family, the pollen grain is shed at 2-cell stage. The further male gamete formation takes place once these pollen grains lands on the stigma. So once this landing takes place, pollen grains are successful. There on the stigma, further development takes place and the generative cell there divides to produce 2 male gametes. But in 40%, everything is done within the anther, within their home. Correct? So till here, development of pollen grain is also known as in situ development. In 60% of the families, 2-cell stage is in situ. In 40% of the families, 3-cell stage of pollen grain is in situ development, because this development is taking place in their home, in their anther. Now anther will burst, releasing all the pollen grains outside. Now as and when pollen grains will get the time, will land on stigma, they will further show development, they will further germinate, and that all will be away from their home on some new place. So that development will be ex situ. But till here it is in situ development. I hope till here pollen grain male gametophyte is clear to each and every one of you. If yes, then it is done. Male gamete is ready. Now let's try to ready the female gamete. For that, you have to study the female reproductive organ, which is called as gynoecium or pistil. Gynoecium beta, it is a collective term for carpels. What is carpel? You know, it is an essential whorl which is present innermost in a flower, right? Structurally, this is the thalamus, right? On the thalamus, carpel will be born, right? Structurally, this carpel, a female reproductive structure, consists of 3 parts. The topmost is stigma. Beta, stigma represents the landing place of pollen grains. The pollen grains, they're going to land on stigma. So it represents the landing place of pollen grain. The swollen base is known as ovary. Yes, the swollen base, you heard it correct, is known as ovary, which consists of ovule. The other term for ovule is Megasporangium. The number of ovule, beta, may be 1, may be more than 1. Correct? Now, these two structures, ovary and stigma, they are connected with the pipe-like structure called style. Correct? Style may be hollow or may be filled with tissues. Such a style which is filled with tissues is known as solid. So what is solid style? Style with transmission. Which are these tissues? These are transmission tissues. Correct? Have you all understood this? The structure of female reproductive organ, that is your carpel, 3 structures, stigma, style and ovary. Ovary encloses the megasporangium, which is ovule. Whole structure is diploid, 2N. Now, there are 2 conditions of carpel. If suppose in a flower, more than 1 carpel is there, that means multicarpellary condition. Suppose in a flower, more than 1 carpel is there, multicarpellary, and they are fused. All are fused to appear as one. Then this condition is known as syncarpus, as you see in case of Papaver somniferum. However, the other condition is multicarpellary, but all are free. You can count the number of carpels in a flower. So that is known as apocarpus. Right? And apocarpus you see in case of mycelia. So just remember these 2 points. Stigma, style, ovary. Stigma is the landing place. Ovary is the swollen base, which after fertilization transforms into fruit, which encloses megasporangium called ovule, that later on transforms into seed after fertilization. There is a connecting tissue called style, which can be either hollow or solid. Now, in a flower, there can be 1 carpel, there can be many carpel. 1 carpel means monocarpal. Multiple carpel means multiple carpellary condition. Now, if there are more than 1 carpel present, they all can be fused. Syncarpus like papaver. They can be free. Apocarpus like mycelia. I hope all the information which is given on the screen is clear to each and every one of you. Yes? Shall I proceed further? Yes, ma'am. Beta, you have understood the structure. You know what is female reproductive organ. But our aim is to see that where is the female gamete. So female gamete, beta, develops inside the ovule, within the megasporangium. Like in pollen, like in androecium, the main structure, the main male gamete develop in the anther. So you studied the structure of anther. Here we are going to study the structure of ovule, which is megasporangium, because within ovule, female gamete is going to develop. Right? Now see, I'll first draw the structure of ovule and then each part we are going to study separately. So this I am drawing the most common ovule, which is inverted ovule, called anatropous ovule, right? First, let me draw this. So I'm drawing actually anatropous ovule, which is inverted and most common. 80% of the angiospermic families, they have this type of ovule. Correct? First, just let me draw it and then I will explain you. Till then, you can just wait and watch the diagram and try to recall your own information and see if ma'am is drawing it correctly or not. If you find any trouble, please wait. So this is the parts of ovule. Beta, this is inverted ovule. Means the things which you're seeing below are actually above and the things which you're seeing above are actually below. That means chalaza that you see at the top is actually the base of the ovule. So first part which you should know is chalaza. Chalaza represents beta base of the ovule, which you actually see on the top. It is actually the base. Why so? Because this ovule which I have drawn is inverted anatropous. Micropyle, the opening of the ovule through which pollen tube will enter. The opening which guides the entry of pollen tube is known as micropyle. It is actually present at the top, but you can see it down as it is again an inverted ovule. So second labeling which you should know is micropyle. It represents the opening of ovule that guides the entry of pollen tube inside the embryo sac. Correct? Now, one structure you see by the name funicle beta. Funicle is the stalk with which the ovule is attached to the placenta. Now, coming back to this diagram, here you can see ovary and ovule. Beta, there is one more structure by the name placenta. Placenta is a cushion-like structure in the ovary on which ovules are born. So, ovules are attached to placenta with the help of the stalk called funicle. So, third structure which you should know is your funicle. So, beta, just see. Suppose this is ovary. Okay, this is placenta, this is funicle, and these are the ovule. So what is funicle? Stalk with which ovules are attached to placenta. Correct? Yes or no? Now, this point of attachment, the point of attachment of funicle to the ovule, this point of attachment beta is known as hilum. So, this point of attachment is known as hilum. Right? Clear to everybody? The first three labelings along with the terminology hilum. Now, coming inside the ovule, the covering of the ovule you see is known as integument. So next term is integuments. Integument represents the covering of the ovule. Usually there are two integuments. In some there can be one integument, and in some there are no integuments. Bitegmic, unitegmic, ategmic. So what are integuments? These are the covering of ovule, which can be either 2, 1 or absent. 2 integuments, like the most common ones have 2 integuments. You can see I have drawn 2 integuments only. One inner, one outer. So this condition is known as bitegmic, most common. So there is one outer integument and there is one inner integument. Bacha, when ovule transforms into seed, these integuments form the seed coat. Correct? The outer integument develop into testa and inner develops into tegumen. Correct? One integument means unitegmic and absent means no integuments. So condition will be ategmic. Clear? Yes or no? Which on fertilization develops into seed coat. Understood this point? One note point I am writing, beta. In case of lychee, there is 3 integuments. In case of lychee, instead of 2, there are 3 integument. The third integument is called as aryle, and that aryle is the only edible part which you eat actually, sorry. In lychee, there are in total 3 integuments, correct? Comma. The third one is known as aryle, and actually it is the edible part. This aryle is only edible. Remaining is seed which you throw out. The edible portion which you eat in lychee is the third integument, which is aryle. Understood? Okay. Now these integuments, beta, they encloses a nutritive tissue called nucellus. So can you see this in the diagram? This is all nucellus. So next point is, just a second, let me add one page. Yes, nucellus. What is nucellus, beta? It is the nutritive tissue which provides nutrition to the developing female gametophyte, which is embryo sac. Right? So it's a nutritive tissue enclosed within the integuments. Right? Now there are 2 types of nucellus. Crassinucellate condition and second is tenuinucellate condition. Crassi means well developed, beta. Some angiospermic families in their ovule, they have well developed nucellus. That is called as crassinucellate. Some, they have poorly developed nucellus, not that much richly deposited nucellus. So it is tenuinucellate. Crassi means well developed nucellus. Correct? Tenui means poorly developed nucellus. Correct? Now beta, when I'll teach you megasporogenesis, then I'll tell you that actually from the cell of nucellus only your female gametophyte embryo sac develops. So next structure, which is actually very, very important within the ovule, is embryo sac, a haploid structure called female gametophyte. Coming back to the diagram, see, this is the female gametophyte. I'm not going to tell you right now the details of embryo sac, but when I'll teach you megasporogenesis, there we will do the details of your embryo sac. But let me tell you, embryo sac is the haploid structure within the diploid ovule that develops after meiosis, and it is the structure which carries the female gamete, which is egg. Hence, embryo sac is the female gametophyte. Why it is known as female gametophyte, students? Because it carries female gamete. Therefore, fertilization inside the ovule takes place in the embryo sac. So you can say that embryo sac is also the site of fertilization. Correct or not? Understood the entire structure of ovule? Is each and every point clear? Whatever I have drawn, whatever I have written so far? Yes, 100%, no doubt. So we are going to keep it till here. In our next class, we will start with types of ovule. See, I have only taught you one type of ovule, which is anatropous. There are many more types of ovule, 5 more types of ovule, which I will teach you in my next class. In our next class, we will study types of ovule and megasporogenesis. So these 2 topics will which we are going to cover in our next class. Right? But before I say thank you, I have some list of homework which I want you all to do and write the answer in the comment section. So there are again 2 homework questions, 2 slides of homework which I expect you all to do, and I really think and I really hope you are doing it seriously. So yes, thank you student for watching the lecture. Keep connected, keep in touch, and yes, be regular, be consistent. Thank you everyone. Bye bye. Take care.